Hello, people. How y'all doing? All right. Well, if you all answered me in unison, I didn't hear you. I don't know. I might have my speaker off or something. Volk, did you get a haircut? I think what happened is I washed it and it just looked like that afterwards. Eggman. You can ask anything you want. That was my car. That's my Dodge Charger. Reading guide optional. I don't know. Is it? Is it chapter 12 reading guide optional? I have no idea. You tell me. I like your cut. I don't know what that means. Eggman. I'm allowed to. Okay. All right. I think I covered all the questions. Brew. Ha. All right. I think we got everything covered. Good. I'm glad I was able to answer all those questions for you. Looks like we're ready for the test with all of that new knowledge. All right. Can everybody hear me? Yes, no, maybe so. How about you guys say something so I make sure my speakers work?
Yes.
Ah, that worked. Excellent. Good. That makes me feel better. Righty. Don't forget we have a test next time we meet. It's on chapter 6, 7, 11, and 12. It's the cells test.
What's this test on then?
What?
You said that we had a test. Today?
Not today, on when we come back from Thanksgiving. We still have to finish chapter 12. We haven't talked about regulation of cell cycle at all. I even posted this in Google Classroom too. I said it, posted it in Google Classroom. What's going on here?
I know, but the way you worded it, I thought you meant today. I was really confused.
So did you study for a test today?
No, that's why I asked.
Oh, I thought just now you thought that. Just now you thought, I thought you thought it for the last week and you've been studying for nothing or something, you're going to be mad or whatever, but I understand now. No, what I said is the next time we meet, we're going to have a test. The next time we meet, so that's on Tuesday, okay? 1 week from today. I believe it's December 1st or something like that. Sam Buckner has made that reading guide optional, the chapter 12. It is now optional because Sam Buckner made it optional. It's in the chat, so it's indisputable. I think it should be mandatory because I think a lot of people aren't going to do it, and I already made one of the other ones optional. I'm thinking 2 of them should be mandatory. So what did I, wait a minute. What did I make optional already? I don't remember. Anyway, whatever. I do just walk through the questions. Every other reading is optional. Okay, well, did I make you do 11? Signal transduction, because that's a super important one. By the way, I just looked through my test. I made the test. It's done. I think it's done. I hope it is. In the multiple choice questions, most of them, not more than half, but like the largest amount of questions is in chapter 11. The second largest is, I think, chapter 7 with diffusion or mitosis. And then the least questions are chapter 6 with cells. Just so you know what, like it's not all equal. You know what I mean? And I also didn't put as many free response on this time because last time you guys were being dinguses and you weren't finishing on time. Even though you had plenty of time, I think it's just because people like take naps in the middle of the test and oh, I'm just gonna go watch some Netflix in the middle of it, take a break. I don't know, but anyway, so yeah, I don't know, whatever, we'll figure it out. What I'd like to do is go through the rest of chapter 12 with regulation of cell cycle and some discussions about cancer, because cancer ties in directly with mitosis. If things go wrong with the cell cycle with mitosis, you know, we got to bring up cancer. So we're going to talk about that. And then I don't know how long that's going to take. That might take us the whole hour, but if it doesn't, I made 2 free response practice questions that'll prep you for Tuesday, and it's not so much the content, because you already know the content that's going to be on there. I mean, you know, you got to study it. You know, there's going to be the signal transduction stuff, you know, water potential, all that. So it's not so much the content, it's more like the mechanics of it. Because the way I'm doing it on the free responses, I'm giving you a Google Doc that has like pictures of like the cells on it. It's going to be one that has like the water potential apple cell thing or potato cell, whatever. And I want you to draw on it with Cami, and that might be tricky. Or I want people to draw with pencil and paper and then upload it with their camera. So I want you to do the practice thing. We'll do that at the end if we have time. And if not, I'll just assign it to you to do like tonight or whatever. And then we can practice like doing that. And if anybody has problems with it, you can tell me before Tuesday. So that on Tuesday, we don't go, oh, well, it's not working, or, you know, I don't know how to do this or whatever. Anyway, okay. Sophia says she has a question. Go ahead. Allie Hampel finished The Office. Nice job. You should start over now and watch again. So Sophia, you can type in a direct message or you can say it out loud, like whatever, but I'm going to start with the spiel now. Oh, before I start too, I want to show you what I put on Google Classroom now today. So I'm just going to show you that real quick. Quickly, sorry, it's an adverb. So first of all, I put these practice questions up here. Okay, so it's actually like the test. It's a Google form. You know, we have to submit the files. So it's exactly going to be like the test, except I changed the questions. Yeah, anyway, so that we'll look at later. And then the other thing I put up is these review terms. So I've given you a giant pile of free response questions to study from. It's got like 30 questions or whatever. So I gave you those like a week ago. I'm sure people are at least halfway done with those. You got another week to finish them. I will not collect them, but you'd be a fool not to do them and not at least to look at them and like make sure you know all of them. Because, you know, if you're like, oh, I'm just going to use my notes and I'm just going to look stuff up on Google during the test, I'm just going to cheat. Yeah, with limited time, that's tricky. That's a tricky proposition. And I would hope that you have enough moral, you know, character to not cheat anyway or not plan on it. All right, review terms. So here's like all the topics that are going to be on here, whether it's multiple choice and free response. I crossed a couple out, but most of them are on here. And so this last part right here, like starting with G1 checkpoints right here, that's what we're going to cover today. So it's really not all that much more to cover, but why don't we dive in so we don't run out of time. Okay, yeah, Henrietta Lacks, that poor lady, my goodness. Feel bad for her. So we got up to here. We talked about the stages of mitosis. We talked about what happens in each stage. And yeah, so there's going to be a boob coming up here. So we'll cover that later. You're gonna have to handle it. You're gonna have to handle it. You're gonna have to control yourself and deal with it. Especially since the topic is a somber topic. It's talking about cancer. So we're not going to make jokes about that. But anyway, so we finished up with the stages of mitosis. We talked about the evolution of mitosis, and we said that dinoflagellates and diatoms have different sort of in-between mechanisms where the nucleus stays intact, and they use different strategies. And we said the diatoms and the yeasts, they use something that's closest to what we have to true mitosis. We said the dinoflagellates were more, even more bizarre, with holes in the nucleus and the chromosomes not even being attached to spindle fibers. Super weird. So that's these kind of weird algae called dinoflagellates. And then diatoms and yeast actually have the chromosomes attached to spindle fibers just like we do, but the nucleus still stays whole the whole time and it just splits in two. And then obviously in modern mitosis, normal mitosis, which most organisms use, the nucleus disappears completely, and then the chromosomes like spread out, and you know, in the middle of the cell they line up, and then they get divided up, which works best. And the reason why that is because more complex organisms have a lot of chromosomes, and you wouldn't be able to do it the primitive way with nucleus is too small for them all to line up correctly and then get separated. So when you have, you know, 46 chromosomes like a human, you're gonna need to get rid of the nucleus to get this done correctly, and that's what most organisms do. And by the way, when I say most organisms, it's a very safe thing to say, because You might say, well, you don't know how many species of these algae there are. Maybe there's more of them, or maybe there's more bacteria than, let's say people and dogs and pine trees and stuff like that. And the answer to that challenge is easy, because the most numerous species on Earth are the insects. They think there's over like 2 million species of insects, or maybe way, way more than that. And only maybe about 1 million discovered. So since there's more species of insects than anything else, even bacteria, even yeasts, even amoebas and all of this stuff, it's a safe thing to say most creatures on Earth, or most organisms, do this kind of mitosis, because insects are most creatures on Earth. So that's kind of a nice little fail-safe there. All right, let's talk about regulation. So I'll go to display mode here. Hopefully this will work. And the faces are gone. No, they're not. Oh, Claire's here. Oh my God, I didn't let her in. Oh, geez. Here we go. And I don't think I ever took attendance. Did I ever take attendance? I don't think I did. Did I? Oh, yes, I did. Yes, I did. Yes, I did. Claire's here. Okay, well, we'll see if she gets in. All right. Every time I do this, I feel like I'm a big rookie here, like I don't know what I'm doing. I don't know if you get that feeling too. She's still waiting. All right, so basically what I'm trying to tell you is the cell cycle, even though it looks like a clock, it's not actually just like a giant clock. It doesn't just keep ticking along, always continuously doing cell cycle stuff. That's not how things work. So a lot of times the cell cycle has to be turned off and turned on when needed. So clearly regulation is necessary. Clearly regulation is necessary. All right, so some of the evidence for that came from some old experiments where they actually fused cells. And if you take a cell, like any kind of cell, mouse cell or whatever it is, that's in the S stage. Okay, so that's synthesis. That's when DNA gets duplicated in the cell cycle. And then you take another cell that's in G1, or maybe like even like, could even be in G0. And you fuse them, the cell that's in G1 that isn't really doing much of anything yet immediately goes to synthesis. It immediately starts duplicating the DNA. It skips all the rest of G1 and it instantly enters synthesis stage. So it's almost like there's little chemical signals or something floating around in the cell that are telling the cell, oh, now it's time to do this. Now it's time to do this. And those chemical signals is what we're going to be talking about in this part of the chapter, which is pretty interesting. And that's going to be new stuff from freshman year. We never talked about this. So that's all going to be new. All right, another experiment, they just took a cell that was undergoing mitosis. So it's, doing like the prophase, all of that stuff, metaphase, anaphase, telophase stuff. And the one that's shown there is maybe metaphase or whatever. And then they took another one that's in G1. So just maybe, close to interphase or not much happening there, nucleus is there, all that stuff. And the cell instantly then entered mitosis, even though it had not duplicated its DNA yet. Like it skipped S stage altogether. So there must have been some kind of chemical signal in that cytoplasm when it fused that told the other cell, go into mitosis. So the nucleus started to break down, all of that stuff. And it wasn't even, ready. It wasn't even ready for that because it didn't have the DNA duplicated. So that all that tells us is that there is something that regulates it. It doesn't tell us what it is, but it's telling us that there is something that can control the cell cycle and mitosis so that it's not just continuously happening all the time, which is what some people think, and that's a misunderstanding. Everybody clear on that? Okay. All right, so we should look at what are the things that are controlling it then, which we shall. So one sort of generic way to describe it is that the cell cycle has checkpoints, which are kind of like traffic lights that either say go or stop or go. Some people even call it stop and go lights. But anyway, we know of at least 3 checkpoints. We know there's one in G1 before S, which is the most important one. And that kind of makes sense. Ideally, you'd want to stop this thing if you want to turn it off before you duplicate the DNA. We know there's another one that's pretty well studied at G2 or at the end of G2, right before mitosis. And then there's another one in mitosis, like around, like right before metaphase or right during metaphase-ish. So why would it be a good idea to stop, if you're going to turn off mitosis and you want the cell to go into interphase in G0 and do its thing, like it's done with mitosis, why is it a good idea to stop it at the G1 checkpoint versus, let's say, oh, just wait till G2 and just stop it then and let it go into G0? If you waited till G2, what would be true about the cell? What would have just happened to the cell? Just go back on the clock and look what's up above the G2 that starts with S. Shout it in the mic. Come on, I want to hear this.
Synthesis.
Synthesis would have happened. So how does it, who cares? So what if there's synthesis? Big deal. Does that hurt the cell at all?
I mean, there would just be extra DNA floating around.
Yeah, so there's tons of extra chromosomes now. We have double the chromosomes. Could that be a problem? In the nucleus having double, like 92 chromosomes instead of 46. Would that be okay or not? Michelle Certain did a head shake, no, but only one. Like she didn't go all the way back this way. She only went like this far. So she was not committed all the way to like follow it through to this side. But at least I like the effort that at least you began the head shake, no, which is somewhat of a response. Somewhat weak, but at least it's something. Anyway. All right, so we don't want to duplicate because literally what will have to happen is if you duplicate the chromosomes and then you don't do mitosis, that cell will die. That cell will have to be destroyed. It will not be a functional cell. How would we destroy that cell? What mechanism could we use? Shout it in the mic. I want to hear it.
Apoptosis.
Apoptosis, exactly right. And by the way, if you pronounce it apoptosis, that's okay. For the last 12 years, I've been pronouncing it apoptosis. I only recently switched my pronunciation. It's still hard for me to commit to the new one. Anyway, all right. So we got these checkpoints, and we're going to talk about even though the G1 is more important, the G2 checkpoint is actually more thoroughly studied. That's the one we understand the best. So that's the one we're going to study. That's the one we're going to use as our example. But keep in mind the G1 is similar. Might be a little bit more complicated, but it uses similar mechanisms, and we're going to study the G2 checkpoint. All right. This just says that the cell will go into G0 up here. So that's kind of obvious that it's kind of nice that the cell can go into G0 from this point, because that's what you want. You want the cell to do its normal activity here. You want it to be a healthy, normal cell at this point. You don't want to have it go past S and then be a messed up cell with double chromosomes. And from here, you can't go into G0 anymore. It's going to have to be destroyed. If any of these two checkpoints are the ones that are going to stop it, that cell is dead. I mean, it's going to die. It can't recover from that. So it can't just like undo all the chromosomes or whatever. You know, just kidding. I'm going to put them back. No, too late. All right. This is just the traffic light analogy. There's really not much information here other than that. So let's talk about the G2 checkpoint in a little bit more detail. The way that works is it uses a special kind of enzyme called a cyclin-dependent kinase. So cyclin-dependent kinase, we just abbreviate CDK. CDK. So CDK is kind of a commonly used in biology abbreviation for cyclin-dependent kinase. I'd like you to know what it stands for, but it's perfectly acceptable to talk about CDKs. It's an accepted acronym. Okay. And obviously, because they're cyclin-dependent, cyclin then is the thing that controls the CDK. The other thing that's kind of nice is that we already know what a kinase is. So can somebody tell me what's a kinase? It's an enzyme that does what?
Adds a phosphate.
Adds a phosphate with the help ATP. That's exactly right. Okay, so it phosphorylates things to activate, you know, maybe other enzymes or molecules or whatever. All right, MPF is just a fancy word or term, maturation promoting factor, or MPF. It's just a fancy term for the cyclin-dependent kinase at the G2 checkpoint. So it's the cyclin-dependent, oops, that's not even a C, cyclin-dependent kinase plus its cyclin makes MPF. Okay, and the way we sometimes draw that is we draw the cyclin-dependent kinase like it has this little receptor point, you know, like an active site almost. And then we have a cyclin coming in, and the cyclin might look like this. Thank you, PowerPoint, for glitching on me and making my job more difficult and causing me tremendous stress. There's the cyclin. Okay, here's the cyclin-dependent kinase. You can see this is going to fit in there. And when the two of them are connected together like this, that whole thing is then MPF. And that whole thing is then going to promote mitosis. It's going to turn on mitosis. When you don't have MPF together, the cyclin and the CDK connected, mitosis will be turned off and the G2 checkpoint will be a red light and it'll say, stop right here, you can't get through. Kind of like Gandalf on the bridge in the mountain. You shall not pass. Do you remember that scene? Okay, what was the scary thing that was attacking them? Name it. Let's see how good you are.
Voldemort.
Oh my God, wrong movie. Holy crap, it was a Balrog. Don't you guys know Lord of the Rings? Disappointing, disappointing. You shall not pass, G2 checkpoint, Gandalf, bam. Anyway, okay, this just shows what I was just talking about here. So we got our CDK right here. That's inactive because there's no cyclin bound. We got cyclin right here. That's going to bind to it. And once it's together, CDK and cyclin, we call it MPF. You know, that's kind of complicated and almost like unnecessary extra names, but it is the way it is. It's biology, sorry. It's the reason I have a job. Iron Golem is wrong, and the Grinch is wrong, and that Voldemort is wrong. You guys are dumb. All right, so that gets it past the G2 checkpoint. It gets it into mitosis. And then in the middle of mitosis, somewhere along the way, maybe during anaphase, CDK actually breaks down the cyclin, and the cyclin falls apart. It's in fragments now over here, no longer active. And then over this period, if the cells need to divide more, cyclin gets rebuilt. So we're going to rebuild cyclin here, and that's controlled by other cellular mechanisms, like, for example, RTKs. RTKs, we know, turn on mitosis frequently. We know growth factors are often controlled by, often use RTKs as their receptor. So that's one of the ways that a growth factor, an RTK, is going to turn on mitosis, is by causing the production of cyclin, which then is able to turn on the CDK again. But it's nice that this has its own off switch built in, that it automatically shuts down so it doesn't keep mitosis active all the time. Because if this cyclin right here would never break down, mitosis would be on permanently, and then we'd have a lot more cells than we should be making. And what's that called? If you make too many cells, we have a name for that. Starts with
Cancer.
T. You say it louder? If you said cancer, that's correct. But what's that mass of cell in a cancer called?
Tumor.
Tumor. Somebody said tumor. Yeah, that's correct. That's correct. Very good. Anyway, speaking of your stupid chat comments with all the mixing up of the stories, I recently was talking to somebody about, was that in this class where I talked about Star Wars and Mr. Spock? Was that you guys? It was like, I'm a huge Star Wars nerd. Mr. Spock's my favorite. Yeah. I was telling my wife, I was so proud of that joke, and I was telling my wife I made that joke in school, and then she was like, they're going to know you did that on purpose, and they're going to think it's really stupid. And I'm like, oh, yeah, she's right. Anyway, yeah, so what else am I missing here? Okay, cyclin and CDK necessary. Oh, CDK never goes away. CDK is always there. It's stable. That protein just hangs around, and it's either going to be turned on with cyclin, or it's going to be turned off when the cyclin breaks down. And if you want, no, she's actually pretty nice. I mean, she puts up with me, so I got to appreciate what I have there with her. So what am I saying here? Okay, so the thing that gets regulated is cyclin. You can either turn mitosis on by having cyclin produced, or you can turn it off by shutting down cyclin production, because CDK all by itself will not activate the G2 checkpoint. It'll get stopped there, and it'll turn off. This is kind of like a fail-safe way to stop mitosis. This is not the main way, because the main way needs to happen at G1. Really, you want to shut it off here first, But if something goes wrong with this and the cell goes, into mitosis when it shouldn't be, you can shut it down here again and here again. So this is more like a backup fail-safe mechanism. Anyway, any questions on this? This is kind of complicated, but not as bad as chapter 11 stuff. Everybody good here? All you people with the camera off, can you turn it on and just kind of then I can see how people are doing by your face and your hand gestures. And by the way, none of you guys were ever this shy in class. You're always, real willing to act like idiots and, make fun of me and talk to other people. And now all of a sudden you're shy. You can't fool me. Like Sim, for example, Sim has never been shy a day in his life. Are you kidding me? And he's the only one with the camera off. Anyway, yeah, you guys literally, it's been what, 6 months and you've forgot how to function like humans. It's pretty depressing. Anyway, if you could turn your face off in real life. That's a good one. I'm going to steal that line. That's a good one. All right. This just shows evidence, scientific experiments they did that measured kinase activity, increased, it spiked before mitosis spiked. That just shows that cyclin-dependent kinase got activated before mitosis. This is kind of a boring graph. This shows that cyclin is directly related to MPF activity. So clearly cyclin is a part of it, that cyclin has to spike before MPF. That's all this shows. That it's basically, this is just supposed to be evidence that what I just told you is true. But since you guys always blindly believe anything I tell you, we don't even need to show you the evidence because you're just going to accept anything I say. If I say the sky is green, you're going to write it down. You're going to answer that on the test and, whatever. So why am I showing you evidence? All right. What does MPF do exactly? How does MPF help mitosis? So let's take a look at some of the mechanisms that it's engaged in to do this. So it's clearly a kinase and it activates other kinases. Okay, that's really generic. That doesn't really tell us much. But the next one tells us something. It phosphorylates nuclear lamina proteins. So the nuclear lamina are those keratin intermediate filaments that make up the inside wall of the nucleus, the nuclear envelope, and it makes it real strong. The phosphorylation of those nuclear lamina proteins breaks down the nucleus. Okay, so it's going to disintegrate the nucleus. It's going to help do that. It's got a role in that. It also has a role in chromosome condensation. Keep in mind, condensation means rolling, tightening up, coiling up the DNA and making the chromosomes into those little sausages, which we know only form right before mitosis. Okay, so it'll help, coil up the DNA into these little tight chromosome sausages and gets the cell ready for mitosis because it has to happen before the cell divides. The chromosomes, the DNA can't be all over the nucleus, all spread out, and then expect it, to divide equally amongst the two daughter cells. It's not gonna work. Volk, answer me what? I don't even know what the question is. What stops mitosis at phase G1? I forgot. Oh, it's another mechanism similar to this MPF, but it's not MPF. It's another kind of CDK. And it's not as well studied. We don't know as much about it. So that's why I'm talking about the G2 one. Anyway, I'm going to ignore the rest of the dumb chat. MPF breaks down its own cyclin. We already talked about that. And then clearly other mechanisms also regulate the cell cycle. MPF is not the only one. It's just the one that we know the most about, that we've studied the most. And this is kind of weird for kids to learn stuff like this, where it's like, oh, wait, why don't you just tell us everything? Well, a lot of these things we talk about, we don't know everything. Like scientists still don't know all the mechanisms of the cell and of living organisms. There's still new stuff to discover. And that's why you people need to, take AP Bio and go to college and get smart and then become doctors and scientists and discover new things, Anyway, so, all right, so that's some of the things that MPF does. And, oh, that would make a nice multiple choice question, wouldn't it? Yes. Or maybe even a free response. All right, there are other factors that regulate cell cycle, that regulate mitosis, other than just the MPF and the CDK stuff. For example, here's one. If during metaphase or, approaching metaphase, if the chromosomes are not all connected to kinetochore fibers. So here we have metaphase. Here we have these kinetochore spindle fibers that are supposed to connect the chromosomes at metaphase. If, let's say, one of them is not connected, so there's a spindle fiber here, but it didn't connect. It didn't grab this one. The cell has a way of telling that, and it's not a CDK mechanism. It uses a different mechanism, but the cell knows, and it will shut down mitosis. It'll freeze mitosis at that point, and it's not going to continue. So it'll wait until this is connected, or if it never connects, the cell will break down, and the cell will do apoptosis and disintegrate. But so that's an example of an internal signal other than MPF that regulates the cell cycle. So all of the kinetochore fibers, all the spindle fibers have to be connected to a chromosome or mitosis cannot proceed, which is good because if this was allowed to divide, you would get one cell with 3 chromatids, you would get the other cell with 1, 2, 3, and then these two would still be together because this entire X right here would go this way. And over here would get nothing. I shouldn't have done an X here because it looks like a chromosome, but this would be, an unequal division. And this cell would be messed up because it has extra DNA. This cell would be messed up because it doesn't have enough DNA. They would both be defective. And this would be a big problem. All right. Oh, sorry, I didn't talk about the bottom part yet. External signals. So one example of an external signal we already talked about quite a bit. We talked about growth factors. We started first mentioning growth factors when we talked about RTKs. Now, if you're sitting here going, oh yeah, RTK, that's receptor tyrosine kinase. That's the one that's 2 monomers that combine to form a dimer and then activate 6 spots where there's tyrosines, which turn into kinases, which then, activate relay proteins to activate some kind of cellular mechanism that tends to be a broad response. If that's what was going through your mind, hey, you're in pretty good shape. If I say RTK and you're going, what? What's that? Well, you might want to study chapter 11 a little bit more. You might want to know what that is because, that's one of the big ones that we talked about. We talked about G proteins. We talked about RTKs. We talked about ion channels. You should know those. Wait a minute, that was chapter 11. Why are you still talking about that? That's because it's all tied together. It relates to chapter 12. It relates to mitosis. RTKs are activated by growth factors. And one example is platelet-derived growth factor. That's a growth factor that has a job to help with wound healing. It helps us heal. So let's say you get a scratch or a cut on your arm and capillaries are damaged. The lining of the blood vessel is damaged and blood is leaking out. You're going to have to fix that. Well, one way the body fixes that is with clotting. Another way it fixes it is it activates mitosis in fibroblasts. And fibroblasts are little cells that can grow and heal up and seal up that hole in the blood vessel. So the fibroblasts will do mitosis and grow and start that closing up process of the injury. Does that make sense? To help heal the blood vessel so that blood doesn't leak out anymore. And that can happen pretty quickly. Like that, it's a nice little system that we have. Platelet-derived growth factor does this. The growth factor is released by platelets, which are the tiny little things, the tiny little fragmented cells in the blood that cause clotting. So it all has to do with that wound healing, blood clotting, starting mitosis to heal the injury and close up that blood vessel so that the person doesn't, sustain a much worse injury by, losing blood internally. Anyway, scabs are actually something slightly different. That's on the outside. So what I'm describing is true tissue. Like this actually quality cells of the blood vessel lining that will fix that blood vessel for real. A scab is like dead white blood cells and stuff like that. That's eventually going to fall off. That's not permanent tissue. That's going to stay there. Yeah, like dried blood, white blood cells that were there for the infection, skin cells that got dried up, stuff like that. Blood clot medicine affect those cells. No, blood clot medicine acts in a different place in the clotting. Blood clotting is one of the most complicated things in the body. It has like 39 steps and it's super complex and I can't even teach it to you because I don't remember it from college. And the blood clotting drugs that people take, like anti-clotting stuff like heparin and things like that, warfarin and all that, those act at one of those cascade of events at one of those steps in the blood clotting mechanism, but I don't think it's at the fibroblasts. I'm relatively certain. Okay, so here's just an example that they got cells on a Petri dish. They got fibroblasts on the Petri dish there on agar, and they on the blue one on the left, they don't give it PDGF, which is the platelet-derived growth factor, and on the right, they gave it PDGF. And what do you know? They started to divide and they started to grow, which is evidence that, yes, indeed, PDGF can activate mitosis. And we know that it's because it activates RTKs, and then activate a cascade of events that will increase cyclin, okay? And the cyclin then helps with MPF and all that other stuff. That's not the only thing it does. That's not enough because you still have to get past the G1 checkpoint, but it's part of the process that you have to get through all the checkpoints. Otherwise, mitosis shall not happen. All right, there are other there are other external factors that we can talk about. And some of these I talked about freshman year. So kids that had regular bio, maybe you talked about this with your teacher, maybe you didn't. If you didn't, pay attention closely. Normal healthy cells will exhibit a behavior that's called density-dependent inhibition. And all that means is that when cells are growing, they're doing mitosis. And if they bump into another cell, like let's say we're talking about those fibroblasts healing a blood vessel where there's a hole. You know, let's say the arm is injured. There's a big cut here, and the fibroblasts are growing back together. When they touch each other, they stop growing. Mitosis gets turned off. Okay, that's called density-dependent inhibition. So contact with another cell triggers inhibition of division, even with growth factors present. So even if there's a growth factor, this will override that and shut it down. The mechanism of this, you don't need to know. And what that means is that I don't know it either, because I didn't feel like looking it up, so I'm not going to make you know it. So don't ask me. So that's one. And this is good because if cells are touching each other and they're done growing and the job is finished, we don't want them to keep growing because if they keep growing, they might make a tumor. They might cause cancer. We don't want that. We want it to be shut off. So that's a nice thing that normal healthy cells do, density-dependent inhibition. Density is like how many there are. And when there's so many and there's a lot or too many, it stops. That kind of makes sense. So the title kind of tells you, the name of the behavior tells you what it does. The other one also tells you anchorage dependence is another behavior that's exhibited by healthy cells. And that just means that cells want to be stuck to a surface. They want to be stuck to something else, maybe another cell. So there's receptors on the outside of the cell on the membrane and part of the sort of extracellular matrix where when they connect, the cell then basically is told it's okay to divide, it's okay to do mitosis. So I'm not saying they're surrounded by cells on all sides because then density-dependent inhibition would stop it. But I'm talking about just in one place it's stuck and then it can grow in the other direction still. That's anchorage dependence. If the cell breaks free and it's freely floating in the bloodstream or in the extracellular fluid, it will stop mitosis. So those receptors on the outer surface of the cell will tell it, nope, we lost contact. You can no longer do mitosis. It'll get shut down. Okay, and this works via those membrane receptors and then being connected to the cytoskeleton on the inside. And once again, that exact mechanism we're not going to worry about. But we should understand anchorage dependence means cells have to be attached. Density-dependent inhibition is when cells are like bumping into other cells from growing too much. They will stop mitosis. All right. That's a little bit confusing because it's almost like contradictory, the two. One is saying, yeah, they have to be bumping. They have to be attached to something, touching something. The other one says when they touch, they stop. But it's like it's a matter of how much touching there is. How much is too much? And when all the cells are being surrounded by other cells and bumping into other cells in all directions, then mitosis gets shut down. I talked really fast and I talked really long. And now I need you to tell me if you understood that and if you have a question. Ryan Nowak, how you doing? Angie, how you doing? I heard you, Ryan. Thank you for responding. Angie, you there?
Yeah.
Excellent. Anna Hoagland?
Yeah.
Wouldn't this be easier if you had your camera on? I wouldn't have to call you up by name.
No, my dog is being my dog.
My dog ate my camera. That's my excuse. All right, cool. This just shows what I was just talking about. And, you know, over here, basically things have gone horribly wrong. The density-dependent inhibition is no longer shutting down mitosis. The cells are growing on top of each other instead of just staying next to each other, like here, the healthy cells here. Unhealthy cells here, when they're growing on top of each other, they're going to form a tumor. Okay, and that's going to end up maybe possibly being cancer then. Cancer cells do not exhibit density-dependent inhibition nor anchorage dependence. They violate both of those conditions. They violate both of those behaviors, which is what makes them cancer. And that means when cancer cells break free, they can keep dividing. They don't need to be attached to a surface, which really is bad for us, because if they stop dividing and if they just got killed off by the immune system, we won't have to worry. But if these rogue cells break free and they continue to divide in the bloodstream, and then they lodge someplace else, like in the lymph nodes or in another tissue, another organ, then the cancer is spreading from the first tumor to another place. So cancer cells cheat. They don't follow the laws of biology. They cheat. To say that they do not respond normally to the body's control mechanisms is it's putting it mildly. First of all, cancer cells may not even need a growth factor in order to do mitosis. In other words, some of them make their own growth factor, which then is released and binds to their RTKs and then turns them on. So it's like they cheat. It's kind of like you writing yourself a note that gives you permission to go someplace or whatever. You wrote the note. It's supposed to be your mom writing the note. Your mom is the growth factor, you know, and you're writing your own note, and now you're, you know, going on a field trip you're not even supposed to be going on or whatever. Who knows? Oh, that's a good analogy. I just came up with that. That's pretty good. That's not bad. You should write that down. It's pretty good. So they make their own growth factor. Some cancer cells don't even need a growth factor. They don't even bother making their own growth factor. Their RTKs are turned on permanently. So the RTKs doesn't even have anything bound in those two cups. You know, like in case you forgot what an RTK looked like. It's these guys in the membrane. So there's not even a ligand bound here, and it's still active down here. It's still turned on over here and doing its thing. So that's cheating. You know, it's supposed to have ligands bound up here that are then going to, you know, activate this. And when the ligands dissociate, when they go away, it's supposed to be turned off. With cancer cells, it just stays on. So that's pretty bad. Abnormal cell cycle control system. Yeah, no crap. That's super obvious. So obviously when the cell cycle is messed up and it just keeps going when it's supposed to stop, that's when you get those tumors. That's when you get cancer. Transformation is when a normal healthy cell actually changes into a cancer cell. So a whole bunch of mutations have to happen. We'll talk about that in later chapters when we talk about DNA mutations and genetics. We're going to bring this up again. So we're going to revisit this again. So about 5 or 6 mutations have to happen for it to turn into a cancer cell. We just call that transformation. If tumor cells stay where they are and they do not metastasize, that's the word for spreading, then they're called benign. So a benign tumor is not as bad as a malignant tumor. Benign means it's not spreading and it's not actually cancer. It's a tumor, but it's not cancer. It's only cancer if it wants to spread, and it's going to end up spreading eventually. That's basically one of the definitions of it. If that's the case, if it is cancer, we call it malignant. And the word mal means bad. Okay, you know that already, like in Spanish, mal or whatever. Okay. So if it's malignant, it's the bad kind that wants to spread, and it will metastasize. And that's one of those funny English words that's pronounced strangely. It's like metastasis, but for some reason we pronounce it metastasis. But you know, I don't know why they do that, but that's metastasizing is spreading, you know, of the cells somewhere else and then causing a new tumor, a new cancer in another location, which is what makes cancer so dangerous and what makes it so deadly. And why it's so important to catch it early. Okay, let me read the thing here. Why are certain cancers caused by different things? Just failure of density dependent inhibition, failure of apoptosis, failure of mitosis. Oh yeah. No, wait. So okay, I thought you said why. Yes, there's lots of different things that can cause cancer. Anything that in the end affects mitosis being turned on from all those different directions could lead to cancer. Okay, like yeah, like if cells are dividing out of control and apoptosis takes care of them, no cancer, no problem. It's not going to be a tumor because apoptosis killed those cells. But if apoptosis fails and the cells are growing out of control, then you might get a tumor. Does that make sense? All the types of cancers affect mitosis? No. Mitosis affects cancers, not cancers affects mitosis. It's the other way around. It's the failure of mitosis to be regulated correctly causes cancer, not the other way around. It's like flies don't cause garbage. Garbage causes flies. Does that make sense? Maybe that's a bad analogy, but whatever, you know. You said that cancers can make their own growth factor. This causes transformation of the healthy cell. Not necessarily. That's maybe one part of it. But like I said, there has to be like 5 or 6 mutations. If a lot of the bad mutations happen together in one cell, then it will transform into a cancer cell. So if just 1 or 2 things go wrong, you might just get a tumor or like a cyst. You might have heard, like somebody might have a growth that's discovered by a doctor and then they do a biopsy and it comes back and they say, oh, it's benign. So there was a tumor there. It's a tumor, but it wasn't cancer because only maybe 1 or 2 mutations happened. Does that make sense? So just the growth factor thing isn't enough, maybe. A bunch of things have to happen for it to be cancer, which is a good thing. So it's not like we're constantly getting cancer all the time, which would be bad. Or there's certain things that need to happen in mitosis for specific cancers. Correct. And it's usually it's always mitosis not being turned off. If mitosis gets shut down too much, it's the opposite of cancer. Then it's like wasting diseases, like whatever it might be, where cells are dying and you're not getting replaced, not getting repaired. That's bad too. So mitosis, you want mitosis to be well balanced. You want it to be just right. You want it to be on when it's supposed to be on. You want it to be off when it's supposed to be off. Too much, cancer. Not enough, then you get, you know, degenerative diseases where your cells start dying, and which could be, you know, just as deadly or worse even. Okay, metastasizing. Okay, here's the breast cancer picture that people got so excited about. Please don't get worked up. It just shows, it's really not that complicated. It just shows how cells in one area of the body ended up mutating more and more and more, ended up growing irregularly. Eventually cells break off and then start spreading through the blood. When they start spreading through the blood or the lymph, they end up getting trapped in the lymph nodes. I don't know if you guys know what lymph is. Lymph is like the fluid that leaks out of the capillaries, then it gets returned back to the veins before it goes to the heart, the vena cava. So lymph is basically the water that leaks out of the blood that then gets returned back into the blood, so your blood doesn't get all, you know, too thick or whatever. And the lymph nodes are along the path of the lymph ducts, and they filter the blood, and there's white blood cells in there that take out bad stuff. So cancer cells will frequently get trapped in the lymph nodes, and that's why doctors always check the lymph nodes to see if somebody's got, you know, spreading or metastasizing going on. So here the cells then got stuck somewhere else, somewhere, you know, not in the breast where it started, and causing a new tumor somewhere else. And this is, once again, makes it super important to get checked out, to go to the doctor regularly, to get checked out. For women, mammograms are important. I know those are horrible and it's not fun and whatever, but it's super important that you catch it early, because if you catch it early, it's very treatable. If you never go in and you don't get it checked, it's more likely that the cancer is going to end up harming you badly. Somebody's just popping in. Let me just check again. Hold on. Sophia got booted. Okay, she's coming back. Okay. All right, some more facts about cancer cells. They have abnormal chromosome numbers. So they're just rogue, weird, bizarre zombie cells. They have weird chromosome numbers. Mitosis is going crazy on them. They don't have the normal 46. They're just weird in every way. Their cell surface changes allow them to detach and then float through the blood or lymph and go somewhere else and metastasize. all right, which normal cells wouldn't do. Normal cells, if they break free accidentally, they would just die and they wouldn't be able to do mitosis, but cancer cells can because they cheat. This one is scary to me. They secrete signal molecules to cause vascularization of the tumor. Does anybody know what that means and why that's scary?
Doesn't it have to do something like the tumor somehow becomes like invincible? Like to the body?
Sort of, but I was specifically asking about this word vascularization. Does anybody know what that means? What's the vascular system? What is my vascular system? Yeah, it's the heart. And what does the heart pump the blood through?
The veins.
The arteries and veins, right? So my vascular system is the tubes that the blood goes through, my blood vessels. That's what we're talking about. So if you have, now we talked about surface area to volume ratio in cells in chapter 6. Oh my God, chapter 6 is popping up again. That's cool. We said that every cell has a blood vessel next to it. It has a capillary next to it to give it oxygen and glucose and take away CO2 and waste and all that. Every cell has that. Every cell needs that because that's why cells are small, because big cells, diffusion takes too long and blah, 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 blah. Well, now you got a tumor, you got your blood vessel here. Now you got a tumor where the cells are growing on top of each other. You got this giant tumor thing. Cells are just growing, growing, growing. Well, guess what? That cell is really far away from the blood vessel. It should starve now. So the tumor should be self-regulating. Like it shouldn't grow very much because it can't get enough food from the capillary. So it should starve and it should die. So then the tumor would never actually cause a problem. Well, but that's not the case. These cancers actually release molecules that cause more blood vessels to grow throughout the tumor. So now your tumor has blood vessels all over the place, giving it, you know what I mean? Giving it nutrients and taking away waste and whatever, and supplying the tumor with what it needs, which is bad for us. It's great for the tumor. It's terrible for us. And it's scary to me because it's almost like this tumor has this like mission, you know, it's like, what? How did this evolve that a tumor makes its own vasculature? Like what? So to me, that's kind of scary. And I'm not trying to like get you guys to be all freaked out right before you break and, oh my God, I'm going to die of cancer and like all that. Like, and that's not the point here, but I need you to understand that cancer is serious business. And we need people to study it. We need people to, you know, to take these classes and to go to college and to get smart and to become doctors and do the research and all of that stuff. So anyway, kind of like, you know, all these smart people that did all this and then were able to come up with the COVID vaccine super fast, which is like faster than we expected. They came up with it in at least two different vaccines that are very promising. So this is like, hey, I'm glad there's smart people out there doing this, and I'm glad there's people that are taking the time to study and to become smart and do the research. And maybe that's going to be you in the future. Maybe you're going to discover something new, you know? That'd be kind of nice. We all evolved from tumors. No, I don't think that's true. The tumor is not a natural, normal part of the organism. So I'm going to disagree on that one. So weight mutation cancer, like does the mutation cause for like formation of proteins and stuff to be altered causing the cancer? Mutations that affect the cell cycle specifically is what I'm talking about. And we'll talk more about that in like chapter, I think 18 or one of those chapters. We'll go into much more detail. So if you can wait a few more weeks on that, we'll get into that. But yes, there's different kinds of proteins we're going to discuss that are actually quite interesting. P53 and RAS and BRCR and all kinds of cool stuff we're going to talk about later. Right, but not for this test. Decreased ability to repair DNA. So cancer cells cannot repair their own DNA very well, so they end up with a lot of mutations and weird chromosome numbers and stuff like that. But that's actually maybe good because radiation can kill them. Radiation therapy can kill them because radiation damages the DNA. And normal cells, healthy cells, can repair their DNA. They can fix it. We'll talk more about that in chapter 16. And cancer cells aren't very good at it. So radiation kills cancer cells more readily than healthy cells, which is nice for us. Chemotherapy is another treatment that we have. So chemotherapy are basically chemicals or drugs that interfere with the cell cycle, but only cells that are doing mitosis are going to be affected by this. Okay, so these are drugs that only affect cells that are actively engaged in the cell cycle. Any cell that's in G0 is not going to be affected by it. All right, and one example of a chemotherapy drug is Taxol that happens to freeze the mitotic spindle. In other words, it stops microtubule depolymerization. So if we're talking about the spindle fibers or kinetochore fibers pulling in this direction or pulling in this direction, it stops these from pulling. It stops the microtubule from getting shorter. So it gets stuck here, and then it's frozen in this position, and then the cell ends up dying. Okay, so that's kind of a nice system. So cells, like regular cells in your body that are not doing mitosis, will not be affected by the chemotherapy drug. You're like, oh, then why are there side effects? Chemotherapy, we know, has some pretty, bad side effects that are very unpleasant. Let's think about the side effects of chemotherapy. There are three main ones I'm thinking about. There's probably more, but there's three main ones. One real common, well-known one is people throw up and they feel nauseated from chemotherapy. Does that ring a bell? They get really nauseated from the chemotherapy. So the reason that happens is because, guess what? The lining of your intestines and your stomach and your just intestinal tract, your GI tract, is generally always doing what? You guys remember doing the cheek cell lab? And I told you, don't worry about scraping away the cheek cells because they're just going to grow back because the lining of the inside of your, digestive system just keeps growing back. So those cells in the lining of your small intestines and stomach and stuff like that, they're doing mitosis. So those cells are affected. So that would make sense that it would cause upset stomach and nausea and vomiting. That's why chemotherapy causes that. Does that make sense? This is the part where you go, oh, yeah, that makes sense. I'm waiting. That's it? Wow. Ryan, can you give me a appreciative noise?
Wow.
That's pretty good. That's pretty good. Hey, Ryan. You ever watch The Office?
Yeah.
So you know how like Michael and Ryan have this relationship like this, they're like, Michael thinks they're best friends and stuff?
Yeah.
Going forward, can we treat our class like The Office where I'm Michael and you're Ryan? And I treat you the way that Michael treats Ryan? Can we do that?
Sure.
Okay. Get ready. This part will not be pleasant for you, but it'll be fun for me. Anyway, okay, another side effect is hair falls out. Well, guess what hair follicles are always doing? It rhymes with schmitosis.
Mitosis.
Hey, so they're doing mitosis. So if chemotherapy drugs affect those cells and kill some of those cells, if you're killing cells in the hair follicles, it kind of makes sense that the hair would fall out. Does that make sense? It kind of makes sense. Does that make sense? Yeah. Okay. Well, Volk, learn how to talk. And then finally, your immune cells are white blood cells, and white blood cells are made in the bone marrow. They're always getting produced in those bone marrow cells continuously by mitosis. So there's cells in your bone marrow that are always doing mitosis and producing white blood cells to help you defend yourself against, infection and stuff like that. Well, guess what? Some of those cells are hurt by chemotherapy drugs, which means when you're undergoing chemotherapy, your immune system is now
Weaker
than normal. It's weaker than normal. So people that are undergoing chemotherapy, you need to be careful not to go around people that have COVID, not to go around people that have the flu or, a cold or whatever, because now they're more compromised and they're more susceptible to getting sick and having, serious consequences from viruses like that or bacteria. All right, so that kind of explains the side effects of chemotherapy, I think, pretty well. Anybody have questions on that? I read this in a book last year. Nice, 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 nice. So radiation is the other approach that we use. So chemotherapy is one, radiation is the other one. Sometimes they do both to treat people with cancer. It depends on the cancer. For some, one is better, for them, the other. For some, they use both. And once again, cancer cells don't repair DNA very efficiently. So radiation, I'm not sure if you know this or whatever, but radiation in DNA causes thymines to bind together. So where the thymines are normally stuck to an adenine like this is what it's normally like. If there's two thymines next to each other and there's radiation hitting them, it causes these hydrogen bonds to break over here, and it causes the two thymines to link together. So it's going to end up looking like this. Now the two thymines are stuck together, and it forms this bulge in the DNA, and the poor little A's over here are all by themselves. They don't have anything to connect to anymore. So we call that a thymine dimer. That's one of the ways that radiation damages DNA. Regular old cells can fix that. They cut out those thymines and they put new ones in, and the DNA is fixed and the gene is perfectly fine. Cancer cells can't fix it. So when you cause a bunch of thymine dimers in cancer cells, the cancer cell ends up dying. That's how you can kill a cancer with radiation. So that's kind of an awesome system that we have. If you do radiation too much, can this hurt your normal cells? Yes, it can actually cause cancer. Radiation can even cause cancer because we know radiation is one of the risk factors for cancer. So too much is bad, obviously, and too much radiation will just kill your cells. So people that get radiation for a cancer, whether it's breast cancer or something else or whatever, will frequently also get like sunburn on the surface of the skin in that place where the radiation is aimed, because it kills some of the skin cells on the outside as it penetrates through. But they have ways of aiming the beams very carefully, so it just hits the tumor. They're very, very good at aiming the technology is awesome for aiming that thing. So they only try to get the tumor and nothing else. Or maybe some of the cells around the tumor, just in case, there's extra ones broken free or something. Can you do chemo and radiation at the same time? I know they do both for treatments. I don't know if they ever do them at the same, like in the same week or whatever. I think they do one and then they do the other, because it would be too much on the system. I'm not a doctor, so I can't speak for certain, but I'd be willing to bet money that I'm right. And most of the time when I make stuff up, I am right. About 87% of the time. If I just invent things, I'm right. This is one of those times. I'm pretty sure I'm right. They do the one and then they do the other. They don't usually do both at the same time because it would hurt the patient. If any of you have doctors as parents, maybe you could ask them and get back to us. Wait, no, why do cancer cells not have the enzymes that fix errors in DNA? Because cancer cells are messed up. They have a lot of issues with the DNA. There's a lot of problems with them. And they keep dividing and the mistakes keep getting worse with the cancer cell. So the regular mechanisms aren't as good in the cancer cell, which is lucky for us because it allows us to use radiation to fix them. All right, this is the last slide. And how much time we got? We got 15 minutes. Sweet, I actually got through something. Whew. Hey, we could start the new unit. This is just a little bit of like history that relates to cancer. I don't think I'm asking you on the test about Henrietta Lacks, but I think it's worth mentioning. This is a woman that lived early part of the 20th century. So she died in 51 of cancer. And what happened is, without her knowledge, some of the doctors that were treating her kept her cells without even asking her. So back in those days, the rules and all that stuff were much more loose. Like nowadays you have all this, patient privacy and all kinds of laws that are very strictly regulating all this stuff. If you want cells from somebody to study, to do research on, you have to get permission. Well, they never asked her, and they just used them. Well, it turns out that her cancer cells were very useful for research. So the bad thing is that they did this without her permission, and that is kind of mean, and it's rude and would be illegal now. The good thing is, yes, the book, a book about Henrietta Lacks. English teacher did a book talk on this. Awesome. Yes. Some AP bio teachers make their students read the book. I do not. I mean, if you want to read it on your own, feel free. It's interesting stuff. And it'll kind of make you mad too about what they did, how they, I think her family got money later, but much later. It took a long time. Anyway, one thing that's worth pointing out here that relates to the biology a little bit more than the history is that normally cancer cells, normal cells, healthy cells, can only divide about 50 times until they die. And we'll talk more about why that is in chapter 16. It has to do with telomeres. So cancer cells defeat this mechanism. They actually are able to reproduce and do mitosis infinitely. And Henrietta Lacks cells, they're called HeLa cells, are still around now. We still have them in research labs, and you can actually buy them like a school. Like I could buy HeLa cells now to use to do, you know, to look at under the microscope and to do a lab with. I don't know if you guys remember Mr. Merkel. He doesn't work anymore, but he used to actually get HeLa cells and use them for one of his Project Lead the Way classes. So we've learned a lot from her cells, so we should be thankful to Henrietta Lacks. Thanks to her, we have learned a lot about cancer. But the way that the doctors went about it was not very, well, not very appropriate. They should have asked her permission and all that. And her family probably should have gotten paid a lot of money. So she ended up dying of cancer quite a short time after this all happened. And so she never was around to benefit from this fame and all that stuff. But I believe since then, whoever read the book can tell us, did her family get some money now? I think they got some quite a lot of years later. She's gonna hog all the Paige, you're just on fire today. You are just full of wit today. All right, and that's the end. And obviously you always look through these questions. You do that on your own. You don't need me to assign them because you look through them because you want to learn more about material, and you're good students and you're hungry for knowledge. As Miss Way would say, you are eager learners. Let's take a look at this. Why don't you all go here now and look at this Unit 2 test practice FRQs and see if you can upload an answer. Now, why don't you just give me a bogus answer right now? Just write blah, 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 blah for the text. Don't actually answer it, and just make a drawing and then submit it. Just to see if it works, to make sure that you know how to do it on the day of the test. So let's actually look at it real quick. So for the first one, your whole question is here, okay? It's this. I would like you to make a copy of this entire Google Doc. You know, it's water potential, just like you know and love. And then use Chpt to draw on these cells, to draw on these apple slices, and actually to write in the solute potential, to type it in there. Okay, so see if you can do that. Now, if you can't get that to work, then you could draw this with pencil, but that would take a while to draw. So then I don't know what's more convenient, using Chpt and draw it with pencil. Either way, you then got to upload to the, you know, once this is done, make sure you keep track of where you have your file, and then upload it here where it says add file. So you would go to this, go to your Google Drive, and upload it to the question.
We're doing like random answers.
Yeah, you can just put a bogus answer now and then do it for real, like over break. Because you can't do this in 10 minutes. These questions are going to take you like 20 minutes easily to solve. Maybe 30 minutes or whatever. So what I'm trying to say is do it for real as practice, because I'm giving you really similar questions to these two on the test. I'm just changing the stuff a little bit. You know, so the second one is discuss how adenylyl cyclase. Here you could just type into a Google Doc and then paste an image in there like that you drew with pencil, or use Chpt. Either way, I don't care. And then add the file. Okay, so I'm nervous about this causing people complications with the technology, where you know the answer and you know how to do it, but you can't figure out how to do the technical stuff. Although on the last test, I did ask you to submit files also, didn't I? And it worked okay for most people. I just remember the nightmare at the end of the test when people stayed like an extra 30 minutes because they couldn't figure out how to upload. And I don't want that to happen this time. Like this time when the class is over, it's over. Like you've got to be done. So does that make sense? So everybody give it a try now. I want to see if it works. And don't worry about getting the answer right or wrong necessarily. And then we'll be good on Tuesday. All right, some of you are maybe misunderstanding what I wanted. I wanted you to actually use the question that I gave you and like edit that Google Doc, like that water potential one with the stupid cells and all that, and write in there, even if it's wrong, and then submit that. I didn't just want pictures of kitty cats or memes or something, because anybody can attach that. That's not hard. I wanted to see if you guys would have trouble with editing those cells and writing into that image, because on Google Docs, you can't easily do that. That's the problem I wanted to avoid here. So by just putting memes in there or whatever, that doesn't help me. So let's see if some other people are doing better here.
Mr. Volk,
Yeah.
Cammy's not working for me.
All right, then this is a good thing that you're discovering that now, because then between now and next Tuesday, you can find a different solution. Okay.
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 I already found one.
Make it work. Make it work, or use something else. Use Sketchpad, use something. Or draw it out on paper and then, you know, take a picture with your phone, but make sure you know how to do that. Anyway, all right, let's take a break from this now. Let's come back together, please. We only have 2 minutes left. Is there any business we need to take care of before we leave for the weekend here? I can't find the doc. You got to save it in your Google Drive in a place where you're going to be able to find it. And the easiest way sometimes to do that is when you're looking for a document like that and you can't find it because it's somewhere nested in, you know, whatever, is just put it in your main Google Drive directory and rename it 0000 and then whatever the name is, because then it'll be first alphabetically. It'll be at the very top when you're looking for the file to attach. What do I have to do so you can redo them again? Just return? I don't actually know what I have to do so you can do them again. Okay, I'll try that and then email me if it doesn't work or whatever. Or if you're coming up with other technical issues, let me know. Sophie Gruber, do you appreciate my background now? No response. Okay, that's nice. I went through all that work for nothing. Any issues we got to resolve before we leave? What should you do over break? I'm giving you no new homework. I thought about giving you a bunch of stuff to do in the book and a bunch of like, you know, stuff to work ahead. And then I decided, no, I don't want to stress you out because I'm so nice. I want you to work on the free response practice ones I gave you where there was like 30 questions. I want you to maybe go through the reading guide, you know, if it's optional or whatever, but maybe use that to practice the mitosis stuff a little bit more and do the practice test thing and then look through those terms. Please look through the terms and make sure you understand what all the terms are for the multiple choice. Everybody clear? All right. Is anybody coming to TSS? Because I'm kind of really hoping nobody's coming. You can come if you want, but I'll probably be mean. All right, stay on if you have questions or if you want to tell me something. Otherwise, have a good Thanksgiving and I'll see you on Tuesday. Bye.